that even previously unknown microbes might be performing. So there exist several methods for microbiome annotation. I'll just mention a few that are closest to our application. So Kraken has been developed for ultra-fast metagenomic sequence read annotation. So taking reads and breaking them into a fixed size schema, and then investigating these schemas against the database, and then making a decision for each read where it sits on the hierarchy given these schema assignments. So this has the drawback of having to fix the length of the schemas in advance, and then taking this read and breaking it. So there might exist longer continuous matches to the database, but because we're looking at schemas, we may not find those. But it's a very fast way of doing things. As well as for each read, you are utilizing the taxonomy tree to get the label. And another approach is called a kaiju, which can be used for amino acid databases as well. So sequence reads are matched looking for the maximal exact matches in the database. And this can be done in different ways to speed up the finding of the maximal exact matches. But in kaiju, the reference hierarchy, the taxonomy is not utilized. So when you get all your matches, so there might be multiple alternative MEMS, then you have to decide what to do with those. So perhaps a post-processing, like in Kraken, to decide where to finally assign the read. So we have some questions we wanted to investigate in terms of the annotation. So people use a hierarchy of taxonomy, so the names of the organisms. But we wondered one could also use a hierarchy of functions instead of taxons. And indeed, by utilizing something called the KEGG enzyme codes hierarchy, one could use this as a functional annotation hierarchy. So this particular hierarchy labels enzymes in 7 different main branches with a 4-level hierarchy. So from root to leaf, there are 4 different levels. And these annotations can be attached to protein sequences or protein domains like we are doing in this work. So we have a database of proteins labeled with these functional codes, and then we can use those with our sequence annotation. And another question is, could we combine these favorable aspects of Kraken and kaiju, so looking at variable length matching, but combining with this with an annotation hierarchy. So as soon as you find a match, you will immediately see where in this annotation hierarchy that read should be placed. And this indeed we approached by developing this method for indexing the sequence data, and then for each sequence in our database, attaching a label to it that links to the hierarchical annotation database. So this could be done on the nucleotide sequences or protein sequences, or indeed any general strings. One could attach these labels. As an overview of the method, we have a database, we have a reference hierarchy, and Promenade does the read annotation given both of these. And the innovation here is in the Promenade method of combining the labeling with the indexing, as well as the idea of looking at functions in terms of hierarchy. And then as the output, we get functions and pathways. So we get a functional profile for each microbiome sample, and then those can be compared and analyzed and clustered and looked at in different ways. Just a little bit more detail. So here's an example. So in the suffix tree, you would find all these sequences in your database, and here each of them point to, or each branch will point to an annotation in the hierarchy. And your actual database sequence reads, their database sequences are used to construct this index, as well as they have these labels that are matching your hierarchy. So as a result, similarly to Kraken, you will get the node as an answer. So you will not get the individual sequence ID, But in this fast approach, you'll get the functional node ID as the output for each read. And this database index only needs to be accommodating for strings as long as your sequenced reads. So could be in the hundreds of nucleotides or 100 amino acids or so on. And here a little more detail. So you can also find all the details in the paper within the methods and the supplement. So conceptually here we have these taxonomic IDs of our sequences in our database. And for each one, we have the lowest common ancestor, or here we call it the lowest taxonomic unit that where the string is found. And here connecting all the possible strings in our database. And associated with this suffix tree, where these labels then indicate the label in our functional hierarchy. So conceptually, when you traverse the tree for each substring, let's say AA here, you can find the lowest taxonomic unit is 4 in this case. And finally, this is, we explain in the paper how this is done for generalized bidirectional Burrows-Wheeler transform. So then the question is, does this all this work to add these labels actually make a difference in practice? So if we don't have very many repeating sequences, for example, in a database, maybe it doesn't make much of a difference. But indeed, in this case of microbiomes and functional annotation, there could be tens of thousands or even hundreds of thousands of similar sequences in a database. For example, some known pathogens like Salmonella and Enterica, there's tens or hundreds of thousands of sequences. And if you want to add all your proteins or protein domains and genes and genomes from these strains, you may end up with a lot of matches to some queries in your sequence, in your sequencing experiment. And then reporting this one answer instead of all the alternatives will give you computational savings, as well as you are really reducing the redundant information in this case if you only care about the functional nodes. So this is similar in read mapping, where you would get all your alternatives, but here they are kind of condensed to the lowest taxonomic unit. So we experimented on some metatranscriptomic data, and I'll tell you the details, but there up to 2/3 of the reads matched multiple sequences in that they had multiple maximal exact matches, and indeed on average up to 12 matches per read. So there's a quite high number of repetition in the database and in the whatever matches from your sequence experiment. And we compared this with the Kaiju, which also looks for the maximal exact matches, and indeed we took less than half the time because we're not spending time reporting all the alternative hits. To kind of summarize the features of Promenade, so again, we look for the longest possible match, and we do not fix the K. At this point, we look for maximal matches. And then the annotation hierarchy gives you a result at the lowest possible level. So some reads may go to the most specific level, and some may even end up in the root because there's nothing differentiating them, or there's too many matches in different branches of the hierarchy. And we efficiently handle large reference databases and high throughput experiments, and allow scalable, scale-free comparison between sample groups when we use our method Rodeo, for example, for comparing these functional profiles across different experiments that may have different sequencing depths. So there's a question, I'm just going through the question as I speak. So the question is, since we're looking at the exact matches, how sensitive is it to sequencing errors, and how would the mem length vary based on error rate? So that's a great question. I'll address some of those, I think, in my slides. So we did do simulations on variable error rates, and I'll show you how the accuracy changes with increasing error or increasing distance from the database, really. And as well as the average length of the MEMS. I'll show you that on the experimental data as well. So faster reads in our pipeline are translated to amino acids. So you get 6 possible reading frames. So 3 possible codon starting points, both forward and reverse. And then promenade classification can be done with the amino acid database and functional hierarchy like we've done here, or it could be nucleotide and taxonomic hierarchy, or any combination of those. And we get the assignment for each read, and then as a post-processing, we summarize the counts at a fixed level, such as when people look at the genus or species level annotations. Here we can look at different levels of the functional taxonomy. And then finally, we investigated these functional profiles by clustering and looking at the differentially abundant functions and enriched pathways between 2 different conditions. And as the database, we used functionally annotated protein domains from the IBM Functional Genomics platform. And in the paper, it was called Omicsware. So if you're wondering, it is the same thing. So you can access the website and check it out. So this is a database as well as a system to analyze data from hundreds of thousands of genomes annotated taken from public databases and assembled and annotated and carefully curated to get a high quality set of genomes and annotated all the way to the protein domains. And indeed, for the simulations, we took data from these protein domains. So at the time of writing the paper, we took about 12 million protein domains that had these KEGG enzyme code annotations. And then we uniformly sampled the reads from those, getting 1x coverage of the database, and then translated those amino acids to DNA, and then added errors to those sequencing reads. And many of these sequences were actually quite short, so a quarter of them were shorter than 500 nucleotides. So we took this paired end sequencing, but we just took the fragment size as twice the read length in order to capture most of the sequences in the database. So here, getting to the accuracy on the simulated data. So if we have no errors, then the green means things went to the correct node, and some of the reads went to the root, but mostly they went to the originating sequence, or they went to the correct path along from this node to the root, so the lowest taxonomic unit for the read. And with 5% errors, things still look pretty good. So only 1.5% incorrectly assigned, and 7% going to the roots. But when you start adding errors up to 10% or even 30% in some case, 2 things happen. So the number of reads going to the roots start increasing. So this may be due to the matches being shorter as well as less specific, as well as there starts to be an increase in the percentage of errors. So the average mem length for this case of 5% errors for 17 amino acids out of the maximum 41. And this is kind of what the distribution looks like. So we also tried this on a smaller database, which is the gold standard database from the MyPhaser paper. And there the lengths were kind of shorter. So that was a smaller database and a smaller set of genomes and smaller amino acid content. But this is what we got for the functional genomics platform on the simulated data. And here too, 10% of the reads would need this LTU computation with an average of around 15 alternative mems per read. So I'll tell you more about this metatranscriptomic data. So this was from a Nature paper looking at how the human gut microbiome changes with diet. So this was an experiment where people ate a plant-based diet and then a diet with animal-based content, and then looking at the differences. So this was around, on average, 20 million reads per sample from 11 subjects during both arms of the diet. And indeed, we see from these functional profiles how these metatranscriptomes cluster by diet, and we find differentially abundant functions mapping to pathways that are relevant in these diets. And just to give some computational aspects, so the index size of the functional genomics platform was about 60 gigabytes, and in classification on our 72 cores took about 1 minute per sample. So this is the data from the original paper showing the clustering based on their taxonomic annotation. So they had a custom reference of 500 gut bacteria mapped to the reference genomes that they used. They got the counts for each gene and then used this as the profiles. And here's just some details on the participant. So in this paper, they also explored the metagenomic sequencing and other omics. So it was a nice multi-omic data source. So here I'm just highlighting some subjects here. This is the vegetarian sample during the plant-based diet, and here based on the animal-based diet. And here's another sample that clusters with the plant-based diet samples, but in our analysis, we found to be slightly different. So on this experimental data, the maximal exact match length was averaged around 8 amino acids, which is shorter than in our simulations for sure. And actually the fraction of reads with a match of 10 amino acid or longer was only a couple of percent. So here this is on log scale, so it's not take that into consideration. So there was very few reads that were longer than 10 amino acid with their maximal exact matches. And contrasted this to a typical k-mer size that one might use of 31 nucleotides. So around that size, only a very small fraction of the reads would be annotated. And here's also a look at the different level assignments in the taxonomy. So this is the percentage going to root versus levels 1, 2, 3, and then the leaf. So in this data, a small fraction of reads actually mapped in the internal nodes of the hierarchy. But on some other data sets, we see more reads here. But in this case, as you increase the error rates, the fraction of reads going to the leaf starts decreasing, and the metatranscriptomic reads in this aspect looks more like the reads with 30% distance from the database, which maybe reflect what we're actually seeing, because a lot of the microbiome still remains unannotated and uncharacterized. And on this data, indeed, 2/3 of the reads would need the LCU computation. So just to quickly illustrate what we mean by the fixed level analysis. So here, if the sizes correspond to your read counts in the hierarchy, and you want to investigate here the lowest level, then you would take counts from the top and push them down based on the proportion of counts you have in each subtree. And then you end up with this comparable counts at your level, at your desired level. So here we looked at the different level assignments. So going from more general to more specific, one sees that the separation between the diet clusters improves. And here the slider green and red, if that's visible, those are the vegetarian, lifelong vegetarian samples that are, this green especially is an outlier to everything else. And this was also apparent from the taxonomic analysis that some of the microbes there are really in different abundance compared to everything else. And comparing this to the data in the original paper, this cluster of 2 subjects with animal-based diets that are kind of in between the diet, plant-based diet, we also see them as kind of outliers here. But this one sample that was intermixed with the plant-based samples, here it clusters with the animal cluster. And this is also nice to see that they are kind of in balanced clusters. So the greens go together and the red goes together. It's not like a nested clustering, it's a balanced 2 main groups of the clustering. And then I'll tell you how we took these profiles and then looked at the differentially abundant features. So this rodeo method we originally published for transcriptomic, looking at actually plant transcriptomes where the reference genomes were still missing, but we had transcriptomes and we wanted to compare the transcript profiles. And here we would take the each sample and we do a bootstrap iteration of taking the sequencing counts and reassigning them. So if here each is cluster, let's say a microbe, we have a certain number of reads out of the total counts, and we use that as the probability of that organism receiving a read or that function receiving a read, and we reassign the reads and reorder the microbes or the functions based on these new counts, and we repeat this many times, and then we get a distribution for all the features. So then we can do this for each sample to get a distribution for each feature in each sample. And then we can compare samples looking at samples of type 1 versus type 2 by combining all the replicates for each sample type. And then you get a differential score, and we can use a statistical test to rank those features and get the most differentiating ones. So Promenade with the functional genomics platform shows these consistent differences as we saw, and here you're only using the top 30 features. We see the samples clustering based on diet mostly. So these green plant-based samples are very much more similar to each other versus these animal-based, which are more variable. So then using these top features, we mapped each to pathways, which is an existing mapping from functions to pathways. And then we looked at which pathways are enriched in each diet arm. So we saw enriched in the plant-based diet functions, including starch and sucrose metabolism, which is higher compared to the animal-based diet, which makes sense in this plant-rich experiment, as well as in the animal-based diet. Enriched were, among others, purine metabolism, and animal-based foods tend to have higher purine content, so that makes sense as well. So these are the kind of observations we can make based on these functional profiles and start investigating what are the biological processes ongoing. And just to summarize, so Promenade allows efficiently labeling variable size exact matches with annotation hierarchy. And once you get your sequence match, the annotation is instantly found. And we introduced this new application of looking at functional hierarchy for annotation instead of the taxonomic hierarchy. And the difference to the well-known methods Kaiju is that we directly report the lowest possible annotation node in the hierarchy, ignoring all the reporting of the multiple hits. And difference to Kraken is that we look at any length sequence matches, so no need to fix the K in advance. And this can also be used for general purpose hierarchical annotation of strings for other applications as well. And with that, I would like to thank the co-authors and thank the IBM research team, and invite you to check out the paper, the GitHub, and the functional genomics database. Thank you.